Well, hello there, and welcome back to our series of videos on the topic of inheritance. So far we've seen how to draw genetic diagrams and Punnett squares, and to solve simple questions involving genetics. In this video, we're going to take a look at different patterns of inheritance, and we're going to practice more of our genetic diagrams and Punnett squares with that. Now we're going to take a look at a few different types of inheritance patterns. First, we're going to continue with Mendel's dominant recessive pattern, and I'm going to introduce to you the idea of a test cross. Secondly, we're going to look at how sex is determined in humans, and then finally we're going to take a look at what is called codominance, which happens in the inheritance of blood type, and this is very important for our syllabus. So first, Mendel's dominant recessive patterns. I'm going to introduce you the idea of a test cross. So just a quick recap. Now in Mendel's experiments, he took a pure breeding tall pea plant and crossed it with a pure breeding short pea plant. And he found that all the offspring produced were tall. So remember how he explained this. He explained it by saying that the genotype of the pure breeding tall pea plant was homozygous dominant, so capital T, and then the short pea plant was homozygous recessive, small t, and therefore all the offspring were heterozygous, capital T, small t. And because tallness is dominant over shortness, all the F1 generation offspring were tall. Now that throws up a very interesting question. And the question is, how would you tell the difference between a tall plant that has a genotype which is homozygous dominant and a tall plant which is heterozygous? Because both of these actually have the same phenotype, right? They look tall. So how do you differentiate them? Now that's where a test cross comes in handy. So what is a test cross? Well, a test cross is basically a genetic cross. It is when you cross an individual that you are testing. So if I'm interested in what is this individual's genotype, then I will cross this individual with an individual that is homozygous recessive. So for example, I might cross a tall plant that is heterozygous with a short plant that is homozygous recessive, and then I'll look at what is the phenotype of the offspring which is produced, what is the ratio of the offspring produced. So let's take a look at what happens when you perform a test cross on a tall plant that is heterozygous. So here's my tall plant that is heterozygous, and I'm going to cross it with a short plant that is homozygous recessive. So this is a test cross. So the tall parent is able to produce either big T or small t alleles, while the short parent can only provide small t alleles. So the result is their offspring will have these following genotypes. And you will realize that the phenotype, the ratio will be 1:1. Half of the offspring will be tall and half of the offspring will be short. A ratio of 1:1. So this test cross comes in handy because we can use it to differentiate between a tall plant that is homozygous dominant and a tall plant that is heterozygous. So let's say we have these two tall plants. Right? We want to tell the difference between them. We can perform a test cross on each of them. So we'll cross each of them with a short plant that is homozygous recessive. Now, for the heterozygous parent, when I cross it with a short plant, it will produce half tall offspring and half short offspring, as we just saw. So 1:1 ratio. But for the homozygous dominant parent, right, it will produce offspring which are all tall, right? Because it's capital T, cross with small t, all the offspring will be heterozygous and all be tall. So basically, if I have a tall plant, I cross it with a short plant. If all the offspring are tall, that means that the tall parent was homozygous dominant. But if some offspring are tall and some are short, then the tall parent must have been heterozygous. That's how a test cross comes in handy. Okay, so putting it into words now, a test cross can be used to find out whether an individual is homozygous dominant or heterozygous. The expected ratios are if that individual is homozygous dominant, then all his offspring will be the dominant trait. So if the individual was homozygous dominant tall plant, then all the offspring will be tall. But if the tall parent was heterozygous, then we'll expect a 1:1 ratio. Next, we're going to move on to how sex is determined in humans. So sex in humans is actually determined by the sex chromosomes. Right? We know that we have 23 pairs of chromosomes, and one pair of those chromosomes are the sex chromosomes. So in female, the sex chromosomes are X and X. So females have 2 X chromosomes, while males have 1 X chromosome and 1 Y chromosome. And the other 22 chromosome pairs are called the autosomes. So here's a diagram showing you the 23 pairs of chromosomes in a human being. You can see 23 pairs, 1, 2, 3, 4, 5, and so on. These are pairs of homologous chromosomes. Pair number 1 to pair number 22 are called the autosome pairs. So these are autosomes. Well, the last pair of chromosomes are the pair of sex chromosomes. So this here is a female because you can see that it has a pair of 2 X chromosomes. If it was a male, then you see only 1 X chromosome and 1 Y chromosome. So a common question that we can think about, that we might be asked is, what is the probability of a child being a male? We can figure this out by drawing a genetic cross. Here's how we draw it. So for our legend, I'm going to use X to represent X chromosome and Y to represent the Y chromosome. So imagine the mother would have a genotype of XX because she's female, and the father would be a genotype of XY because he's male. So the mother can only produce gametes which have a single X chromosome inside, right? Her 2 X chromosomes will segregate and end up in different gametes. And the father can produce gametes with either X chromosomes or Y chromosome, but not both. So when we combine it in fertilization, combine the gametes together, you'll find that the ratio is 1:1. Half the offspring will be female, while the other half will be male. So the probability of a child being male is 50%. And the probability of a child being female is also 50%. It's an equal chance. But which parent decides the child's sex? Well, if we look at the genetic diagram, you realize that the parent that decides the child's sex is actually the father, right? The sperm, because the sperm is the one that could contain either the X chromosome or the Y chromosome. The mother can only give X chromosome, and that's shown in this diagram here, right? The egg can only have X chromosome, but the sperm could be either X or Y. So if you're a boy or girl and you don't like it, you can blame your father, not your mother. Alright, I'm going to end off this video briefly with this extra section on incomplete dominance. So this is not in your syllabus, but it's just interesting to know. So incomplete dominance came out of a situation where a man decided to cross a flower called the 4 o'clock flowers. So these 4 o'clock flowers come in red variety or white variety. So he took a red one and crossed it with a white one. And I want you to imagine what came out. So if you were Mendel, right, if you're looking at Mendel's results, you might expect either all the flowers to be red or all the flowers to be pink. That would be dominant recessive, right? So let's say all the flowers that came out were red. Then red will be dominant and white will be recessive. But that's not the case, because what he found was that when you cross red and white flowers from the 4 o'clock flowers, you get all pink flowers. So this is an example of incomplete dominance. So what is incomplete dominance? Incomplete dominance occurs when one allele is not completely dominant over the other. So in this case, the red color flower allele, red color, is not completely dominant over white color. And the white color is not completely dominant over red color. So they kind of get a mixed phenotype where they all are pink. So the heterozygous phenotype ends up as a mix of the homozygous phenotypes. And this is an example to show you that Mendel's simple dominant recessive model is limited. Right? Not everything in genetics is simple. One is dominant, the other is recessive. Sometimes there's no clear dominant or recessive one. Right? Sometimes both alleles kind of mix together to get a mixed phenotype. So how can I explain incomplete dominance? How can I explain this pattern of inheritance? I can use the symbol C W to represent allele for white flowers, and CR to represent allele for red flowers. I use C because it means color, and W because it means white. And notice that I don't use the same symbol as like big T or small t, because there's no dominant recessive relationship, right? They're both kind of equal, neither is dominant over the other. So the red parent, the red flowers, the parent's genotype will be CR, CR, right? Both alleles are red, while the white parent will be CW, CW. So the gametes produced, the red parent will only produce gametes with CR, while the white parent will only produce gametes with CW. And therefore, when you combine this, right, all of the F1 generation will have the genotype CR, CW, and therefore all will be pink, which is exactly what we observe. So this is how we can, the genetic diagram we can use to explain incomplete dominance. Okay, that's all for this video, and in the next video, I'm going to talk about co-dominance, and we're going to talk about the inheritance of ABO blood type.